Thank you, Ben. Thank you for the very kind introduction. I'm very happy to participate in this exercise. I don't really know how many people are connected, but you can see my credentials here on the first slide. There's my email address. So let me see if I can turn this into a laser pointer so it's easier to see. So you can see here my email address, and then in the lower right corner, my website. If you have any questions throughout the presentation, please reach out to Ben. And if you would like to informally talk to me after the presentation, feel free to send questions to my email. I'll be happy to address any concerns or comments or questions that you may have. My research program at Hopkins, and like Ben said, I come from a very um, hardcore background in fundamental theoretical electrochemistry. And I remember when I was about to graduate, I had a conversation with my then research mentor, Professor Alan Bard, and I asked him, Dr. Bard, what should I do after I graduate from my PhD here at UT Austin? And Dr. Bard said, well, you should pick something that nobody else is doing. Try to find something that is completely new. And at the time, the only thing I could think of was, well, very few people are working in the space of bioelectrochemistry. And so I wanted to expand my knowledge of biology, my knowledge of biophysics, and then do something at the interface of these disciplines. And so the presentation that you will see today is the product of that postdoctoral training that I followed at the University of California, Santa Barbara, that is now continuing to expand at Johns Hopkins University. The motivation for the work that we do comes from the realization that human biology is very beautiful, but also very diverse. So in this sample of all humankind, which is very limited to my group of friends, you can see many different skin colors, many different backgrounds, different sexes, different eye colors, different ethnicities, different cultural backgrounds. And yet, our approach to pharmacology for the past many decades has been focused on thinking that all of these people have a similar type of metabolism in which if they were to take a medication, let's say a pill, and if you had a tool that will allow you to look at the concentrations of that medication in the bloodstream of these people, you will expect that this population, in average, will present a pharmacological profile that will rise and fall depending on how the medication was given to these people. And the idea is that you can potentially derive useful pharmacological parameters out of this profile, like, for example, the peak concentration in plasma or the time at which we reach that peak concentration after taking a dose. Now, you're all familiar with this concept of, well, if you're sick, you have to take a pill every 8 hours or every 12 hours. Well, what that's doing is we're basically trying to build up the concentration in your plasma until we reach a steady state in which we can hold the therapeutic level within a therapeutic window. But you can also imagine that because we're all different and we all have different metabolism, it's very easy for you to overshoot so that you reach a level where you can have some adverse effects to that medication, or to undershoot and to reach ineffectively low levels, right? And this is not a big problem for things like aspirin. You will have to take a lot of aspirin to get sick, but there are many drugs out there that have a very narrow therapeutic window. That is, the dose that is therapeutically effective it has to be very narrowly controlled. Otherwise, those medications can become very toxic. One example of these molecules is shown here on the right-hand side of this slide. This is a very old anti-seizure drug called phenytoin. And what I'm showing you here is each one of these red dots that you see on the graph are the peak plasma concentration of this medication after it was given to a different person. So each dot represents a different person at a daily dose given by the x-axis. So if I bring your attention to a daily dose of 5 milligrams per kilogram, you can see that to all of these tens or hundreds of people that were given the same dose, there's a dramatic variability in the peak plasma concentration that we get, even though the dose was adjusted relative, for example, body weight or body surface area. And so the reality is that for these types of drugs or medications, we don't have a good way to immediately know what the right dose would be for the specific patient and at what time interval it should be dosed. Instead, what we do is we have to come up with an array of those strengths, and then we have to titrate or carefully increase the dose of the patient until we reach a therapeutic level, and trying to avoid potentially having side effects. And so my thinking is we should be able to do better than to titrate individual patients to reach the right doses. We should have some kind of technology that should allow us to address these from the get-go, so we don't have to experimentally individualize the dose for a patient. Another challenge with the approach that we have been using for drug metabolism in pharmacology for the past several decades is that, you know, if you take a pill every 8 hours, it assumes that your metabolism is not changing over time. But in reality, if you're an active person, if you drink water, or if you, for example, had a really fat, greasy hamburger, or if you're a smoker, or if you drink alcohol, all of these changes in your normal daily routine will be affecting the way in which this drug profile behaves and looks like in your blood. And so if you take this as an example, the only parameter I changed was the rate of elimination for each one of these spikes. But you can see that it doesn't look like the ideal profile I showed you earlier, where you had constant intakes that quickly build up to a steady state concentration. In other words, metabolism is not a constant, it changes over time, right? And so the idea that my lab is pursuing is that the development of electrochemical sensing technologies supporting the continuous real-time measurement of specific molecular targets directly within the human body could potentially transform the way in which we study and understand human health, but also the way in which we study, understand, diagnose, and treat human diseases. Such a technology would, for example, enable the measurement of patient-specific drug metabolism or pharmacokinetics, moving us one step closer to the realization of this idea of personalized drug therapy, in which the dosing schedule of a medication is specifically customized to a patient based on actually measured data, as opposed to using crude estimates of the sort of take one capsule by mouth 3 times a day. We can also imagine that such a technology would enable important new medical applications, like, for example, feedback control drug delivery, in which you now have to imagine having a sensor implanted in the body connected to a drug delivery device. And you can use these two devices with a computer algorithm to finely tune the plasma concentration of a medication, so you can control it and keep it within therapeutic levels, potentially avoiding toxic peaks or ineffectively low concentrations. And if we allow ourselves to think big and imagine, close our eyes and imagine the future, we can conceive adapting these technologies to the real-time assessment of health status by monitoring in real time the concentrations of drugs, metabolites, and biomarkers via the use of portable wearable devices, right? So we're not that far away from these technologies. Now, of course, these ideas are not new. We're all familiar with things like the continuous real-time measurement of blood gases. If you have ever been to an ER or to a doctor just to have your oxygen saturation measured, you have had this finger glove put onto you. We're also very good at measuring neurotransmitters in the brains of living, breathing rats, right? And this is via direct electrochemistry, direct detection of monoamines or glutamine, adenosine, or some other neurotransmitters. And we can also detect some metabolites via enzymatic detection, like glucose. So we're all familiar with glucometers or the continuous glucose monitor. We can also measure things like lactate or pyruvate. But the problem with all of these approaches is that they are non-generalizable to the continuous sensing of other arbitrary molecules. In other words, if you want to detect glucose, you need an enzyme that will metabolize glucose to give you electrons, right? And we're not that good at producing enzymes. And so you cannot possibly conceive a scientist producing an enzyme for every single therapeutically relevant drug that we would like to measure. Many drugs that we take are non-redox active and they cannot be detected via direct electrochemistry. And in the case of blood gases, the interaction is based on spectroscopic measurements, which are not readily possible for a vast majority of drugs that are clinically relevant. And so our drive is to produce a general sensing platform that we could deploy in the body to improve the way in which we do real-time continuous drug measurements. I'm getting a little bit of feedback. Are we still good, Ben?
Yeah, you're doing fine.
Okay, great. The platform we're pursuing right now as a first approach is called electrochemical aptamer-based sensors. This technology was initially developed by Kevin Plaxco and co-workers at the University of California, Santa Barbara. The first paper published on this platform dates back to, I think, 2005. Very briefly, I'm going to explain how it works to you. In this cartoon here on the left side, you see an electronic conductor, which in this scheme is represented by a gold wire that we modified with monolayer chemistries or surface passivation or surface blocking chemistries that serve two purposes. The first one is to uh, prevent the occurrence of non-desired electrochemical reactions on the surface of this conductor by blocking it. But also we can use the terminal end groups of these monolayers to customize the chemistry of the device to make it more biocompatible. We also go ahead and functionalize these surfaces with a single-stranded synthetic piece of DNA called an aptamer. Aptamers are short oligos that we select in vitro because they present a very high affinity for a specific molecular target. And lastly, we modify these short oligos with a redox reporter at the end. In most cases, methylene blue, but it can be other molecules. And very importantly, when we expose this interface to the presence of a molecular target of interest, because this aptamer binds to that molecule, there's a strong molecular interaction that changes the secondary conformation of our molecular receptor. And by changing that conformation, we also indirectly change the electron transfer properties of the redox reporter. So the system has two states, an unbound slow electron transferring state and a bound rapid electron transferring state. We can interrogate this interface using many different electroanalytical techniques. The one that is most commonly used is called square wave voltammetry. I'm not going to go into the details of the technique, but suffice it to say that we're applying a voltage program with pulses and we're measuring the current readout. And the output of these measurements is this Gaussian-like curve that in the absence of target has a certain size. Remember, the reporter is directly attached to the interface, so we always have some background current. But the idea is that as you increase the concentration of the analyte of interest, you will see that square wave voltammogram increase in size monotonically with increasing target concentrations. And just to illustrate this point, I'm showing you here an EAB sensor binding to the antibiotic Tobramycin. Each one of these colored dots is a different sensor. We have a batch of 7 sensors. And you can see that they're all responding in the same way to increasing concentrations of the analyte Tobramycin. Now, my lab has spent a lot of time working with these sensors, and one of the big pushes has been to make sure that the community has access to the protocols and methods that we use to fabricate the sensors, but also to software that can be used to interrogate these sensors in real time. And so this is a picture of my student Sam, who's now a first year graduate student in the pharmacology program at Johns Hopkins. And Sam has spent a lot of time coding open source, freely distributable Python-based software that allows the rapid real-time interrogation of these sensors with a temporal resolution of milliseconds. So we can really crank up a lot of measurements as you go. And I'm not going to go into the details of this software, but I can point you in the direction of this paper later on in the presentation. This is a screenshot of what the sensor looks like. Again, I'm not going to go too much into detail, but you can see that we are processing in real time voltammograms at different square wave frequencies and doing some mathematics on them to calculate this percent signal change that in this particular experiment is responding to the injection of the antibiotic Tobramycin following this particular profile. And by doing different kinds of mathematical manipulations in the way we process the data, we can use software to improve the signal to noise in these measurements. One really cool thing about EAB sensors is that because the molecular recognition element, as well as the redox reporter or the signaling element, are coupled to the electronic interface, they readily support real-time continuous measurements in complex biological fluids. This is, you do not need to do any sample preparation steps or batch steps. To illustrate this, I'm showing you this graph here. This experiment was done using an injection system that we have in the lab. The first syringe contains 100% undiluted human serum. The second syringe contains human serum plus molecular of interest, in this particular case, tobramycin again. And the third syringe contains 100% serum again. And so what we do is we're continuously recording every 40 seconds the concentration of tobramycin with the sensors. And then we inject with the second syringe a certain amount of tobramycin. We see the sensor respond. We wash it off. We see the sensor come back down. And we can do that continuously, again, in 100% undiluted human serum for about 12 hours. And you can see that we hit the right concentration of the drug based on the measurement from the sensor with plus or minus 20% error. And in fact, our most recent sensors can do this with a precision better than 20% error. I think we are down to 10%. Depending on the aptamer that you use to fabricate these sensors, the aptamer dictates the sensitivity. So if you have a very good binder that can interact very strongly with a small molecule target, sometimes we can get EAB sensors that will respond to physiologically relevant concentrations of target. And so this is an example for another antibiotic, kanamycin, just showing you the calibration curve in between the dashed lines, the range that will be relevant to humans. And let me point out that these measurements are done. So the points that you see here are the average of 6 sensors plus the standard deviation. And these measurements were performed in 100% whole blood. So we're not diluting the blood or processing it in any way. Because the kinetics of interaction between the small molecule and the receptor are very fast, or in other words, the on and off rates of these affinity-based interactions are very fast, we can perform measurements in real time with outstanding equilibration times. So again, in this example, you're looking at a sensor, a single sensor that is being interrogated continuously. And the moment we spike this pool of whole blood with blood that contains kanamycin, you can see the sensor immediately responding to that stimulus within maybe a minute or less, and then equilibrating that after the injection. And this spike that you see here is the time it takes for us to homogenize the bath of whole blood with this concentration of kanamycin. So the sensors readily support continuous real-time sensing within physiologically relevant changes in concentration. Now, we can make these sensors small enough to fit inside of our animal research subjects. These are rats. So the sensors, so this is an actual photograph of a sensor bundle, which contains a working electrode where we have the aptamer chemistries. We also have a counter electrode that is barely visible in this picture. And right here, you can see another wire. That's the reference electrode, which is a silver chloride reference. Because the diameter of this sensor is about 200 microns, so roughly twice the size of a human hair. So these are very, very small. We have to encase them into medical grade catheters because otherwise they're too malleable and they can bend very easily or flop around in the veins of rats. So we use these medical grade catheters as stents to keep them in place and make sure that we can support continuous measurements. And then we go ahead and do surgery in live animals to place the sensors into a highly irrigated place in their circulatory system, very close to the heart. We have a second line for a catheter that we use to perform drug infusions into the animal. And by achieving these surgical procedures, what we obtain are, baseline corrected, flat, real-time, continuous measurements of, in this example, 2 intravenous bolus infusions of tobramycin. And so you can see the sensor recording measurements in real time. This is the first IV bolus. You see that instantaneous rise in concentration of the drug in the plasma of the animal, followed by first-order elimination kinetics, and then a second bolus injection and first-order elimination kinetics. And I usually like to stop here for a second and remind my audience, this is the first measurement ever performed in situ in the vein of a living, breathing rat that is continuously monitoring the concentration of a small molecule target, tobramycin, without depending on the chemical reactivity of this molecule. Remember, the interactions that these sensors use are just based on affinity, on intermolecular interactions, but there are no chemical reactions happening, which means that this platform is modular. And at least in theory, by simply swapping the molecular recognition element, the aptamer, for an aptamer that binds to other molecular targets, we should be able to use the same platform to create sensors for other molecules that may be clinically relevant. And so in the next slide, I'm showing you a now somewhat outdated panel of the different drugs that we have measured in situ in the veins of living rats. The first three panels in the upper left corner are showing three antibiotics, kanamycin, gentamicin, and tobramycin from the family of the aminoglucosides. We have two cancer-killing drugs, doxorubicin and irinotecan. We also have a glycopeptide antibiotic, vancomycin, and the energy molecule that is very relevant to biological research, adenosine triphosphate. And in all of these panels, I am showing you different types of experiments. This is a titration in vivo to show that the sensor is responding to increasing doses of the drug. These are two serial IV boluses of gentamicin. Because we're sensing molecules directly in the bloodstream of animals, it doesn't matter how we get the drug into the body of the animal. So for tobramycin here, I'm showing you an intramuscular profile. To obtain these, we're injecting the drug into the buttocks of the rat, so into muscle tissue. And so what you see now is a first order kinetics of absorption of the drug, followed by first order elimination kinetics due to the animal processing the drug and excreting it over time. We can do multiple serial IV bolus injections of a drug, in this case, doxorubicin. This is to show reproducibility and sensitivity. And we can do the same kinds of things for other molecules. So really, the goal here was to demonstrate that we could create a generalizable platform for the sensing of a great variety of clinically relevant molecular targets. Now, these are the profiles that we get now, right? This is what, if you were to go to medical school today, this is how you will learn drug metabolism and pharmacokinetics, right? And the reason why this profile look like this with these black points at discrete intervals is because doing any blood analysis up until today require you to restrain your patient to a chair, or in the case of our rats, to a plastic tube, and then draw blood out of the patient or the rat, and then send that blood to a processing facility, right, to do LCMS measurements to quantify the drug in the serum. The contrast is that we can now do this in real time continuously without any sample preparation or sample processing steps. So really taking the whole field of of pharmacokinetic measurements forward to achieve outstanding precision given the number of points that we have in each one of these profiles. So for the rest of the presentation, I just want to cover three important points that I mentioned early in the introduction, and that we have somewhat demonstrated that our platform could be potentially useful for these clinical applications. The first one relates to patient-specific pharmacokinetics, because we have outstanding temporal resolution of, in the left panel here, one measurement point every, I believe, 5 seconds. We can resolve pharmacokinetic phases never seen before. So for the first time, we were able to measure this distribution phase for doxorubicin that has a half-life of 3.6 minutes, right? Think of it. If you were drawing blood out of a patient and then sending that for analysis, you would never be able to achieve this kind of temporal resolution. We're not limited to measuring every 3 seconds. We can go from square wave voltammetry-based measurements to chronoamperometric-based detection using the same sensor interface. And with chronoamperometry, we can integrate the sensors every 300 milliseconds. And so now we have thousands of points in each one of the parts of this pharmacokinetic profile, and we can really resolve the linear infusion, the linear portion of the drug infusion going into the animal. And then this plateau here, which we're now calling the mixing phase, or the time it takes for the drug to reach homogeneous concentrations in the body before the body starts metabolizing and excreting it via the kidneys. We don't really know if these new pharmacokinetic phases, like the distribution phase or the mixing phase, are clinically relevant, but nobody else was able to measure them before. And so we think that we're providing extra parameters that may be useful for the treatment of certain patients in the future. Another important thing about the platform is we can, for the first time, resolve changes in a patient's or in a subject's metabolism over time. So in this slide, I'm showing you two sets of profiles. This is rat number 1, rat number 2 at the bottom. Each rat was given 2 serial IV boluses of tobramycin at the same dose. And what you see invariably between the two is that the first dose has a half-life of elimination that is almost 2 times as fast as the second dose, right? So the half-life here is 32 minutes. The half-life here is 71 minutes. And you may ask, well, why is the metabolism of the animal slowing down and getting, twice as slow as the first injection? And the answer here is very simple. These rats are sedated. They are under anesthesia. They're not drinking water, so they're getting dehydrated over time. Their blood pressure is dropping over time during the surgical procedure. And so we expected their metabolism to slow down as well. But what's important is that our real-time continuous measurements are now allowing us to resolve these pharmacokinetic differences that clearly demonstrate that metabolism is not a constant, right? Now I want to take you to this idea of feedback control drug delivery. So very quickly, the idea is simple. We now have a sensor that supports real-time continuous tracking of molecular targets in vivo. We can couple that to a drug delivery device or a drug pump via a computer algorithm. We're currently using PID controllers. And without going into too much detail, a PID controller is essentially the same type of controller that you have in the oven in your kitchen, right? So you can set your oven to 300 degrees Fahrenheit, and the controller will make sure that you heat that temperature and keeps it constant. We're using the same technology to achieve a real-time control of drug levels in vivo. And so what this graph is showing you is that the sensor is detecting concentrations of the drug in real time. When we turn on the controller, the pump starts infusing drug into the body of the animal. The sensor picks up that dose delivered. It starts to ramp up the signal that we get. And then when we reach a set point that we indicated before the experiment to the controller, then an error is calculated that has a proportional, integral, and derivative parameter that then in turn compute an order to instruct the pump to either stop infusing drug or infuse more drug. And so what you end up getting is a zigzag of the drug levels dropping and increasing again and dropping and increasing again to keep them within the therapeutic level that we assigned for this experiment. And then the moment we shut off that controller, you can see the kidneys of the animal excreting the drug following first order elimination kinetics, as I showed you before. This controller is not limited to a shape or a specific duration of control. So this is just to show you 3 different rats, rat 1, rat 2, rat 3, with 3 different feedback control programs. And you can see the sensors responding in accordance to those feedback control delivered doses. Very importantly, the noise in these measurements is dictated by the noise set by the sensor itself. So it doesn't matter that we're measuring in 3 different rats. You can see that the noise level is basically the same, plus or minus 5 micromolar, because the sensor is the same sensor, it's just placed in 3 different animals. We can also do very long continuous measurements with a constant therapeutic level reached and just holding it there for as long as we can. At UCSB, when we started doing these experiments, the protocol that we had available limited our experiments to basically 6 hours total. And so you will see that all the graphs that I have will be shot before 6 hours. But very importantly, within half an hour, we reach steady state concentrations. And because we know the steady state plasma concentration of the drug, and we know the rate of infusion, we can readily calculate in real time the elimination half-life of the drug over time, right? Or in other words, how the metabolism of this individual is changing over time. And so what you see is that in a period of 5 hours, we have a fluctuation in drug metabolism of about 2 to 3-fold, right? So again, this is just to emphasize in a more quantitative way that metabolism is not a constant, but it's changing in real time with our daily routines. We have done this, so the previous slide is Tobramycin measurements. We have also done this for other drugs. So here I'm just showing you a second example with vancomycin. Again, the same type of profile, holding the dose for about 5 hours, and then monitoring changes in metabolism in real time. This is a slide that I use to give you a point of reference. Because we do not have feedback control drug delivery today in the clinic, the best thing that we can do is something called a constant rate drug infusion. An example of this, for the particular case of tobramycin, you will have a patient sitting in a chair, and the patient will be receiving an IV constant infusion of a dose of 10 milligrams per kilogram at a rate of 10 microliters per minute. And that dose will be delivered over a period of 1 hour. Now, the idea here is that you're infusing the drug very slowly into the vein of the patient, hoping that you will eventually match the rate of elimination from the kidneys to reach a steady state. But what you can see in this example that we collected in rats is that if you have a rat that undergoes a sudden change in metabolism, shown here in red, you can see that blue is the preset PK profile that we're trying to generate. And you can see that we miss in both cases. In one of the rats, we overshot, and in the other rat, we undershot. And that is simply because unless you have some kind of real-time feedback mechanism, you cannot control changes, real-time changes in the metabolism of a subject. The very last thing I want to show you is that these measurements cannot only be made or done in sedated animals, but trying to think about the future, we would like to be doing this in normally behaving subjects, right? And so an example of this was, this is the very first experiment that we ever did with a live rat. The name of this little guy is Charlie. Charlie was our very first test subject. We operated Charlie 2 weeks in advance to implant 2 catheters, one for drug infusions and one for placement of the probe. We let Charlie recover from the surgery, and about 3 weeks later, we came back and implanted a probe into his vein using this leash to protect the probe from him, because he wasn't very happy, as you can imagine. He was trying to pull it off all the time. And this leash is bringing the wires to our potential stat or electronic control. And this is a very typical image of CH software. We now have fancier interfaces with the software that my student developed. But this is just showing that as Charlie's exploring his cage, moving around, drinking water, just freely hanging out, we can continuously perform measurements in real time. And the pharmacokinetic profile that you see on the right-hand side of this slide is the data that came from this experiment. So you can see that even though we have a little bit more noise, we can still follow this profile in a freely moving, behaving rat. And I believe this is the end of my presentation. There are many people to thank. The majority of the work I showed today was done by me and a group of colleagues when I was a postdoc at the University of California, Santa Barbara. However, we are really trying to push this technology forward. And my group at Hopkins is now devoting 100% of our efforts trying to expand the library of molecules that we can use to sense things in vivo, as well as making the sensors last longer in the body so that we can potentially start to use them for real pharmacological studies in humans. And so I'm going to go ahead and thank the students involved in this work currently: Sam Curtis, Alex Shaver, and Miguel. We have funding sources that come from the School of Medicine, as well as private companies and foundations. And really, the heroes of this work are these little guys here, the rats, who let us do all of these experiments on them and are really helping us push the frontier of what can be done with these biomedical devices. And of course, I would like to thank all of you for your attention. I hope this presentation was sufficiently entertaining for a Friday evening at 11:30 p.m.
Thank you very much, Netz. It was absolutely outstanding. Like I had been looking forward to it, and yeah, you delivered. That was really cool.
Thank you. I'm glad that you enjoyed this. It's I hesitate a lot. It's a little late and I'm tired, so hopefully it wasn't too terrible.
No, like I said, that was fantastic. And it looks like okay. So let's see. Infinite 55 asks, after reading through some of your literature, it would seem that a major concern associated with this type of technology is that the bioelectric interface inevitably degrades after a prolonged period of time. How are you addressing this concern, and what was the longest time you were able to achieve?
That's a great question. With the sensors that we currently have, without any modifications, so the original sensors developed by Plaxco ever since 2005, the longest measurements that we have supported in vivo have been about, I would say, 12 hours. But I have to say 12 hours were, the best sensors we ever made. We are now extensively exploring different chemical parameters that we can modify to improve the operation life of these sensors and make it last longer. And I think a part of the concern has to do with the different chemistries that we use that have really not been updated in the last 15 years. So we are changing everything. We're changing the DNA, we're changing the monolayers, we're changing the reporters, we're changing the bonds that are involved in the different parts of the interface. So my lab is doing a lot of chemical synthesis to change monolayer chemistries, the composition of the DNA aptamer, to make it more tolerant to this biological degradation, producing better redox reporters, and also looking into the bond chemistries that we use to make sure that the linkages we have in the interface are not the weak points, basically.
Okay. Very cool.
Did that answer the question?
Yeah, I think so. Looks like somebody's trying to. We got somebody in the chat trying to ask a question, and it's getting auto deleted for some reason. Let's see.
Technology is supposed to help you, but sometimes
yeah, it's supposed to.
Against you.
It's catching a language filter, and I'm not sure why. Holy, you want to try one more time? Oh, there we go. All right. Not sure what was catching the filter there, but okay. So he wanted to, oh, never mind. I'm sorry. It was okay. Holy wanted to say that it was a great and inspiring talk, and was also curious if you think this approach can be used for the analysis of metabolization of radiopharmaceuticals.
As in radio label? Is that? I mean, I think it will be presumptuous to think that you can use a platform to detect every single molecule that could be clinically relevant. I think the EIB sensors that I've been discussing today will be useful for certain molecules that, or for which you can develop an aptamer that presents very high affinity towards that molecule. If you wanted to do something that is a radio label, if I understood the question correctly.
Yeah, he was asking about radio labeled.
You could detect those molecules with an aptamer, but if it's radio labeled, the easiest way to sense those molecules is using a detector for radiation, right? And in that case, the signal is the purest signal that you can get, because if you use the right marker, there's no other source of radiation that you could sense except for the molecule that you're trying to measure.
Okay. Are there any other questions in the chat? Hopefully without tripping a filter by accident. Oh yeah, Holy got a follow-up. All right. Yeah, Holy, go ahead and type up your follow-up. I'll run this other question real quick. So Infinite wants to know, how much potential does this kind of technology have for penetrating or revolutionizing medical diagnostics and the administration of analgesic medications?
Well, that's a great question, and I'm obviously very enthusiastic about this platform. So I'm going to tell you a lot of potential. But being serious here, I think that using this to monitor anesthesia is definitely a high impact application that we're pursuing. We are also trying to develop these devices move a little bit away from continuous sensing and just thinking of single point molecular diagnostics that you can use at the point of care, or even in a decentralized format at home or at a community center. The challenge there is any technology that you want to truly demonstrate to work on the field has to be properly validated. So we currently have two, I shouldn't call them clinical trials, but we have two IRB protocols that are designed for us to properly validate the platform for specific molecular targets. And in these protocols, what we're doing is we're working with the Johns Hopkins Hospital to get actual real patient samples in large numbers that we can cross-validate with whatever the benchmark approach they use at the hospital. So I can give you an example. One of the molecules that you would want to measure could be, and I'm just going to make it up, it could be caffeine. And most likely, the hospital will be detecting caffeine based on immunoassays. So the idea of these clinical studies is that we're going to be getting the same samples from the hospital that the hospital measure via immunoassays. And we're going to compare the output from those immunoassays, which are the gold standard, against the output from our sensors. And we're going to do this with large enough numbers so we can do proper statistical analysis to demonstrate that the platform is accurate, what the precision of the platform is, what the probability of producing a false positive or a false negative errors are. And once we have that information, then I think we can claim to the community, listen, this technology has a lot of potential as demonstrated by these clinical studies that clearly show that it could be used in the clinic. Unfortunately, these studies haven't been done by anyone yet. And so we're really excited to be pushing the boundary in that sense and try to get them done so we can better address the question about potential related to biomedical applications.
Okay. So I got another question here. So what if we have radiopharmaceuticals that bind to different biomarkers, such as myelin and also inflammation? Is that something that you can distinguish with this methodology?
So that's a very good question. I think it's a little complicated given the example of the radio labels, but one thing to keep in mind is that the specificity of the platform relies heavily on the specificity of the aptamer. So we can only be so specific and so sensitive as the aptamer would be for the specific target you want to measure. That being said, there are papers out there of very good groups that have produced aptamers that are able to distinguish between, for example, enantiomers. I wouldn't say that you can generalize this to all enantiomers in the world, but some aptamers can be sufficiently selective to distinguish between two molecules that are only differing by the mirror image they have, right? So that speaks a little bit about the specificity. In real life, though, if you wanted to look at, for example, metabolites, so a parent drug and its metabolite, that metabolite will have to be significantly different, I would say. If we're just talking about methylation, I think an aptamer will have a hard time trying to distinguish those two. Now, if this molecule is binding to two different types of proteins, for example, or two different types of receptors, then the question there is that maybe you should select an aptamer that detects the bound form of the molecule and not the free form of the molecule. So again, it all comes down to the problem, what is it that you're trying to achieve, and how you design your experiment to appropriately address the question that you're trying to answer.
Okay. He meant they gave a specific, I guess it's 18 fluoride or fluoride, FDG. I don't know what FDG is. Maybe you do.
I do know what it is.
Okay.
We don't have an aptamer for that, but it could be measured, I think.
Okay.
I will have to look at the formula and dig deeper into that.
Okay. So are there any other questions from the chat? Because if not, I've got a couple. Or I guess I'll go ahead and ask mine. And if you guys have some more, feel free to ask them. Just a technical question, I guess. You mentioned you actually have a 3-electrode probe.
Mm-hmm.
So how do you, so in the reference was a silver chloride, how do you ensure that the silver chloride stays, how do you ensure that it basically stays good as a reference electrode and doesn't change over time?
That's a great question. I'm going to stop sharing my screen because I have another presentation that I want to show you very quickly. Okay. So,
all right. So I'll hide your window real quick while you do that, and then I'll bring it back up.
Yeah, just give me a second because I have a video that will illustrate this point really, really well.
Okay.
This is a video I used at BitCon, so it should be here. Okay, let's see if I can open this presentation. So the reference electrode is a silver wire that we treat in sodium hypochlorite to produce a chloride film. Okay. This is a very slow oxidation process because it's in dilute solutions, and that ensures that we form a pretty. And we leave it incubated overnight, so it's a pretty thick silver chloride film.
Okay.
And these form very slowly.
Okay.
But let me just show you a quick. So this is the slide. Okay, can you see that?
1 second. Yep. There you go.
So remember, we have so I didn't explain this very well, but we implant the sensor in the right jugular vein of the rat. Okay. We have the drug infusion line going into the left jugular vein of the rat. The screenshot that you see on the right hand side of the slide is this. It's a screenshot of the software that my student Sam developed, right?
Okay.
And what I'm going to show you now is not square wave. It's the interrogation of the sensor using cyclic voltammetry.
Okay.
But you will only be looking at the forward scan. Okay.
Okay.
And I'm going to run this very quickly, right? So each one, you're seeing this blink because that's the real pace at which we're doing these measurements, right? So we're recording all of these measurements, and the software is interrogating the sensor in real time. And what you see here on the image on the right hand side is the progressive decrease of the peak current from the voltammogram over time.
Mm-hmm.
And what you see at the bottom is the position of the maximum peak current voltage.
Sure.
Right?
Okay.
And so even though we have a pseudo reference electrode, you can see that our reference potential is not changing at all. Really.
Yeah.
During so 800 files is several thousand measurements.
Yeah. Okay. So it is actually very stable.
It's very stable.
At least in the time
expanding
frames.
If we're thinking about expanding these to days, then the question remains, is it going to be that stable over time? I think the answer there is we will probably have to use some kind of hydrogel to fix the silver chloride so it doesn't progressively come off over time.
Yeah, I mean, and that seems like something that you could do where as long as you account for, say like additional resistance or whatever, that seems like something that wouldn't be that big of an issue.
Right, and these wires are small. They're about, so each one of the individual wires is 70 microns in diameter.
Okay.
So resistance is less of an issue because we're not using big electrodes.
Right. Yeah. Okay. That's very cool. Okay. So I had another question for you.
Mm-hmm.
So
go ahead.
Early on, you don't have to pull it up again, but you showed a calibration curve. And I don't remember what the molecule was, but it was going from around 10 to the -6 to about 10 to the -2. But it looked like as you got to the higher concentrations, it was almost like there was a little more noise, which you would expect, but it looked like there's some roll off. Do you know why that happens, and is that going to be a hindrance to utilizing these in actual applications?
No, this is a great question, and there's a very simple explanation. The molecular receptors that we use, right? So the single-stranded piece of DNA behave as single binding site receptors, right?
Yeah.
So they follow single binding site physics, which means in an ideal case, they should obey the Langmuir isotherm.
Yeah.
The Langmuir isotherm has a dynamic range of about 1.8 orders of magnitude. and it's sigmoidal in a semi-log axis scale, right? And so what you see here is a sigmoid of that Langmuir isotherm.
Yeah.
So that's where you see the signal plateau.
Yeah.
It should plateau because we only have one binding site. The noise that you see here comes because at the saturation point of the receptor, the majority of the sites on the surface are bound, at least in theory. And so a lot of this noise now becomes very small fluctuations in the receptors as well as non-specific binding.
Okay.
And non-specific binding will be heavily a function of the quality of the surface that you have on that sensor. Okay. Now, addressing your question regarding whether this will be a problem or not, you do not want to be measuring at the top end of this curve. Okay. Because that's where you have the least sensitivity in reality, right? Because you have fully saturated the surface. So ideally, you want to measure close to the KD or the dissociation constant, which is halfway in the rise of this Langmuir isotherm. So about this point here. Okay. So ideally, this sensor will be good for sensing concentrations of tobramycin in the order of 100 micromolar to maybe 1 millimolar. Okay. No, this is 1 millimolar, so I would say maybe 700 micromolar or so. Yeah. So you want to be sensing in this range. Okay.
And I guess you could design the sensor such that I'm assuming you kind of have an idea of what a target range is going to be for a given molecule, right? Or a given medication or whatever it is that you're studying. So you can basically design the sensor and say, all right, we're going to set it such that the upper range, such that it's good up to, say 10 to the minus 3 or whatever, but the range, your toxicity starts at like 10 to the minus 4. Is that a fair assumption?
I mean, Ben, if chemistry was something that you could control, I would say your assumption is absolutely correct.
Yeah, life would be a lot easier.
The reality is, it's very hard to produce binders to target specific sensitivities for binders. Okay. I just wanted to highlight this. This is a review paper that I published with colleagues, and actually you will see Karen's name here. Yeah. We all published this together. We all worked with this platform for specific purposes, all different purposes. My colleague Jorge works from the Air Force Research Lab, so he had more of a defense-focused application for VAB sensors. Okay. Karen has been working for a company called Diagnostic Biochips. They develop probes for in-brain measurements of neurotransmitters and other molecules. Okay. So that's another application for the EV sensors. Yeah. You can see my name, and everything I do is pharmacology. And then Philippe is a colleague who's now back in Canada. He's a professor at University of Sherbrooke. And he is also trying to pursue some of the drug-based detection that I am doing, but also exploring other avenues. In this review, as the title suggests, we were pretty objective and critical, and we laid down all the challenges that the platform has. Okay. With the hope that colleagues will help us address them in a significant way. So for the people in the audience today, and for you, if you like this topic and you're interested in learning more of the challenges, this review talks about the questions that all of you have been asking. Okay. They're all mentioned there in much more detail than I covered today.
That's fantastic.
And if you don't have access to it for some reason, just let me know and I'll be happy to send it to you.
Sounds good. All right, let me think if I have any other questions. If anybody else has them, feel free to get them in now. Yeah, because it looks like the reproducibility was good. You showed a wide range of concentrations, obviously a wide range of molecules themselves. How difficult? Okay, so all right, here this is one I had. So how difficult is it to design the aptamer for let's say somebody comes out with a new drug, they develop a new whether it's a new formulation of an old drug or for an old problem, or if they somebody comes out with I don't know, say a therapeutic treatment for a disease. How hard is it to design the aptamer to work with the sensors like you've described?
So again, I encourage you to go to this review and you can go into much more detail.
Okay.
So the way we choose the aptamers is we use a process called SELECTS. I always forget what the acronym stands for, but it's I think selective. Man, I always forget. This is shameful. It happens to me every time because it's a complicated name.
That's a long acronym.
It's select. It's something like selection of ligands by exponential enrichment, something like that.
Okay.
I should know this, but I always forget.
It's fine.
The way the process works is you start with a library of DNA oligos of different sizes, different lengths, different sequences.
Mm-hmm.
This library is, it can be done in many ways, but one way to do it is you have this library anchored to a surface, some kind of solid support,
mm-hmm.
for example, particles of some kind, and then you have the molecule of interest,
okay.
the molecule that you want to select against these aptamers.
Mm-hmm.
And so you expose the library to that molecule, and if any of the sequences bind to that molecule, they will be released from this solid support. So you can remove the library, collect the sequences that did bind to the analyte, then do sequencing, amplification, and then do it again, and do it again, and do it again. And so typically you go through 6 to 8 rounds of these. Some people do more, some people do 10.
Mm-hmm.
There are all kinds of challenges along the way because depending on how you do it, you may be losing the strong binders that are present at low concentrations at the beginning. But you may also be selecting more of the non-good binders. And so there's all kinds of tricks. It's actually not trivial. And people are now coming up with ways to do non-selects identification of aptamers. I am not very familiar with those. But again, to give you a more satisfying answer, if you check out this review,
mm-hmm.
We have diagrams explaining exactly how it works.
Okay,
and we have a critical discussion of what is it that people don't do with the selection that should be done if you want to then translate these aptamers to the specific application that we have, which is making electrochemical biosensors.
That's fantastic. Okay, yeah, I'll make sure I'll provide a link to that paper when I put the video up, so that way people can be directed to it.
And it really is not. I think you know me well enough, but this is not an effort to self-promote.
Oh yeah, no.
I am very critical of the field because I don't think that my older colleagues are ever sufficiently open about the challenges that we face.
Yeah.
In terms of the platform and the limitations it has.
Yeah.
So I am particularly proud of this work because I think the 4 of us did a very good job at openly, providing a review of what the state of the art is and where it fails, and the things that we have to do to overcome these limitations that we currently have.
Yeah, I mean, that's an important thing to do. If you just act like everything works perfectly, there's no way, how are you going to advance anything? That's right. You have to identify the problems to direct the experiments to try and overcome them. So
absolutely,
that makes perfect sense.